We're going to turn to some, on the skeletal side, some work from Frank Berry, Adipose Stromal Cell Therapy for Osteoarthritis of the Knee, and he'll tell us about clinical progress and challenges.
Thank you very much. I'm very happy to be here, and I'm very appreciative to the Musculoskeletal Committee for giving me the opportunity to talk about the work that we've been involved in. And I'm going to turn very much towards the cell therapy side of the business and very much towards the translational end of that, and tell you about the work we've been doing in developing a clinical trial strategy in Europe for testing stromal cell therapies, specifically for the treatment of osteoarthritis. So the principle of stromal cell therapy is very straightforward. The cells are extracted from a tissue source, which is typically either bone marrow or adipose tissue. The sample of tissue is sent to a facility where the cells are expanded, and then when sufficient cells are available to make a therapeutic dose, they're returned to the clinic and delivered to the patient. And so the strategy is in principle very simple, but the translation of that is in fact very complex, and there's a lot of issues that still need to be addressed. The characteristics of mesenchymal stem cells or mesenchymal stromal cells that we all talk about and which appear to be important in terms of utilizing these cells in a therapeutic context are summarized in this slide. So we know a lot about the surface markers that these cells express, and I'll talk a little bit more about that in a while. We understand something about the multipotential nature of these cells, and we know a lot about the secreted factors that the cells apparently secrete when they're delivered to the host, and which may impact the therapeutic outcome. We also understand quite a bit now about the in vivo niche that these cells occupy specifically within the bone marrow compartment. So the reason for targeting osteoarthritis as a disease to look at the application of cell therapy is very, very obvious. It's because it's a very prevalent condition. It's the major contributor to disability throughout the Western world. It causes a great deal of pain, loss of mobility, loss of employment opportunity, and so on. But despite that, there's no approved intervention of any kind which is disease modifying, and every approach is simply symptomatic and not regenerative. So the case for looking at cell therapy strategies for treatment of osteoarthritis is very, very strong. And there's a large body of preclinical data. We and many others have done a lot of animal studies that show that when you deliver stromal cells as a suspension by direct intra-articular injection into the OA joint, there is a very significant repair response, and multiple models of osteoarthritis in multiple species have shown very, very strongly positive results in this. So the preclinical database is very convincing. And so several years ago, we started a clinical trial in Europe. It was called the Adipose A1 clinical trial, a phase 1b study specifically designed to assess the safety of autologous adipose-derived stromal cell treatment for the treatment of moderate to severe osteoarthritis of the knee. It was an 18-patient study, and when it was completed and the results were looked at, there was a few findings which were interesting. First of all, there was no evidence of any adverse events of any kind. In fact, the safety profile of the treatment seemed to be very good. Secondly, there was an interesting and unexpected pain response in all of the patients that received the cell treatment. Every patient within 24 hours of the intra-articular injection reported a significant and sustained improvement in pain response. That was measured using the VAS score. There was also within 1 week of the treatment, all of the patients that received the cells showed an improvement in functional outcome, the knee function measured using the WOMAC score. So the pain response was interesting and unexpected. And the second observation, which was also unexpected, was that of the 3 doses that were tested in this small study, it was 2 million, 10 million, and 50 million cells per injection. The smallest dose gave the best response. And it seemed that there was an inverse correlation in terms of the therapeutic outcome and the number of cells that were delivered. This was a bit unexpected, but it's since been reported in a number of other studies as well, that the lower the dose, the better the outcome seems to be, at least in these musculoskeletal applications. So that led us then to put in place a strategy for a phase 2b study, and eventually that started in 2015, and now it's possible to report on the progress of that. But before I talk about this particular study, it is worth mentioning that there is a very wide literature describing the use of cell therapy for the treatment of osteoarthritis, and a systematic review, which has just been published in Osteoarthritis and Cartilage, makes some interesting points about the published work. In fact, because of the poor quality of design of the clinical studies, the poor characterization of the cell product, it's really not possible to draw any firm conclusions based on the vast majority of published studies. And in this particular analysis of something like 2,400 patients which have been treated, only about 300 of those represent studies for which we can draw definitive conclusions. And that's, as I said, because of the limited high quality evidence and the lack of long-term follow-up, and the particular issue of the lack of consistency of MSC preparations and the lack of reproducibility of methods used to prepare the cell product. And this issue of consistency of the cell product, I think, comes up time and time again, and it has a strongly negative impact on the field, and it's something that we need to address very strongly. So the Adipose A2 study population then consists of male and female subjects between the ages of 45 and 70 years with mild to moderate knee osteoarthritis. The phase 1 study was moderate to severe OA because that's what the regulators required. For the phase 2B study, they allowed us to look at earlier stage disease. And the particular KL radiographic score that these patients had was between 2 and 3. That's an indication of early disease, and the inclusion and exclusion criteria, the clinical parameters that were applicable, were very carefully worked out. The design of the study then is summarized here. It consists of essentially 3 cohorts of patients, a total of 150, 50 receiving a placebo, 50 receiving a low dose of 2 million cells in a single injection, and 50 receiving a higher dose of 10 million cells in the single injection. And the study is multi-center, so it takes place in 10 hospital sites in 6 different European countries. And in terms of the logistics of all of this, there are 3 manufacturing centers. One is in France, one is in Ireland, and one is in Germany. And so the patient tissue is sent to one of these three facilities. The cells are extracted, and when they're ready, they go back to the clinical site. The clinical management of the trial is summarized here. Essentially, it consists of 9 patient visits. The first visit, the patient comes in for assessment for eligibility, consent, and screening. And if the patient is deemed to be eligible, if the screening results are good, the viral testing is okay, and if the patient consents, then the second visit is for a liposuction. So the patient comes in, undergoes an abdominal liposuction under local anesthetic. A small amount of fat tissue is taken from the abdomen, a minimum of 60 mils of fat, and then that's sent to a processing facility, a GMP processing facility, and the stromal vascular fraction is prepared. The cells are plated and expanded over a period of 2 weeks, and then the cells are harvested as a fresh product, not cryopreserved, packed into a syringe and shipped back to the clinical facility. So the strategy is relatively straightforward, and then there's 7 follow-up visits after that, up to 2 years, where the patients are assessed for pain function and for structural changes in the joint measured by X-ray and by MRI. And at this point, we're just about halfway through the study. Something like 70 patients have been treated so far. And again, as in the phase 1 study, there's no indication of any adverse events, no adverse findings associated with the treatment at this point in time. But it'll be some time before we know the clinical outcome. In terms of the liposuction, we've used a water-assisted body jet apparatus, which makes the liposuction very simple. It's very good for the patient because it's more comfortable for the patient. It's easy for the physician to carry out the procedure, and we think that it reduces the incidence of contamination of the lipoasterate material, which is sometimes a problem in this procedure. So we use this water-assisted liposuction, and then the material goes through a manufacturing process which is very straightforward, and it's summarized here. Essentially, the fat tissue is treated with collagenase and digest, and it's centrifuged, and we get the stromal vascular fraction. The cells are counted and then plated. And over a period of 2 weeks, the cells are expanded. They're passaged once, and on day 14, the cells are harvested then, and as I said, packed and shipped to the clinical site. The overall manufacturing process is summarized here. It's very standard. It depends on adherence as one of the critical parameters for isolating the cells. The release testing includes mycoplasma, endotoxin, microbial sterility, flow cytometry for selected markers, and karyology. So it's all really quite straightforward. Just for those that may have an interest in the economics of this, the cost of an autologous cell preparation is extremely high, and one of the barriers to moving this forward on a large scale is the high cost. So currently in Europe it costs us about 18,000 euros per patient batch for producing this autologous product, and that's really not sustainable on a bigger scale. So that's one of the things we're addressing. We think that we need to bring this cost down to about 25% of what it currently is. So this clinical trial is underway. As I said, it's halfway through in terms of patient recruitment. The hope is that by the end of 2018 or early 2019, all of the patients will have been treated, and then midway through 2019, we'll reach the primary clinical endpoint, which is a 6-month treatment point, and then we'll have the beginnings of an understanding as to whether this treatment is effective or not. And the hope is that the outcome will be such that we'll know in a definitive and unambiguous way whether this treatment works or not, because we really need high quality evidence from carefully controlled clinical trials to determine whether this treatment strategy works or not. But what I wanted to talk about now is, in addition to the need to generate high quality clinical evidence, is the issue of the translational obstacles that still exist in this field, and the serious gaps that need to be filled before we can think about moving this technology forward on a large scale. So some time ago we came up with this translational roadmap, which tries to identify the specific challenges which still exist and which need to be addressed before we can roll this out on a large scale. And so this translational roadmap addresses biological questions and logistical questions, and both of them have very serious aspects which need to be understood. If we look on the biological side, where the biological challenges still exist, chief amongst these is the question of mechanism of action, because we have still a very poor understanding about the therapeutic mechanism of action that's evoked when these cells are delivered to an injured tissue. Years ago, we used to think about sort of tissue engineering principles and the idea that we were replacing new tissue, that we were building new cartilage, and I think it's very clear now that that's not the case, that something else is happening, and it's almost certainly an immunomodulation. But we really need to understand in much more precise terms the therapeutic mechanism of action, and this has to be done for every disease that we're looking at so that we can understand the molecules that are involved in the sort of signaling process between the delivered cells and the host. And until we understand the therapeutic mechanism of action and what the molecular pathways are involved there, we'll never be able to devise effective quantitative potency tests which predict the therapeutic activity of the cell product, because at this point in time we don't have any quantitative tests that predict potency or therapeutic activity. Therefore, we can't really calibrate the dose that we should be using for the patient, and we can't personalize the treatment in any way. So in many ways, we're working blindly because we don't have effective potency tests, we don't have an effective understanding of the mechanism of action, and all of these need to be very carefully understood. What we probably do understand reasonably well is immunomodulation, the way these cells interact with cells of the host immune system, and that's been relatively well characterized, but other aspects of tissue repair and regeneration are not well understood. On the logistical side, we really need better isolation methods because, believe it or not, we still rely on plastic adherence as a major element in isolating these cells. So we need better methods of prospective isolation, we need scalable bioreactor systems, and we need antibody selection, antibody-specific selection methods. We also need to turn completely to xeno-free cell expansion conditions and away from the use of serum and materials of animal origin. So what I'd like to talk about in the remaining few minutes is some of the things we've been doing to try and address these challenges, and specifically how we can understand more about the biology of these cells. And so we've been looking at new markers for MSCs. We've been looking very carefully to try and determine new useful markers that we can use for characterizing these cells. And these markers would then be used as quality release tests for cells for clinical use. So one of the approaches that we've taken is to look at the global surface immunophenotype of expanded MSCs to try and understand which markers could be potentially useful. So to do this, we developed a combinatorial antibody profiling approach, which essentially involves a panel of 230 markers, 230 antibodies which identify surface molecules that can be expressed commonly on cells. We tried to correlate the expression of these markers with the quality of the cells. We did this by taking 3 bone marrow donors. We divided these marrows into 3 and sent them to different laboratories for expansion of the cells. When we did that, we looked at the quality of the cell preps that were made in the different labs and then correlated the expression of different markers with the quality of the cells. So it was essentially a high throughput flow cytometry exercise where we tried to correlate the expression of markers to the quality of the cell preps. And so we then looked at the level of expression of the markers ranging from negative, which is less than 10% of the cells express the markers, right up to high when greater than 85% of the cells express the markers. So we're correlating the prevalence of marker expression in the cell population to the quality of the cells. So we grew the cells, or the different labs that were involved in this exercise grew the cells under different conditions. Condition A was where the cells were grown in 10% serum, but with a relatively high plating density. Condition B, where the cells were also grown in 10% serum, but with a lower plating density. And then condition C, where the cells were grown in a lower serum concentration and with the addition of growth factors. What we observed was that 2 sets of conditions were very good in terms of growth kinetics. When we looked at the growth kinetics under each of these conditions, conditions A and B were very good, but condition C was poor. So this suggested to us that the cells grown under condition C were of lesser quality than the cells grown under conditions A and B. So we therefore tried to look at how the markers that we had selected distributed between these different preparations. What we found is, looking at the 230 markers that I was referring to, is that 10% of them were always positive and therefore not capable of discriminating between the good preps and the bad preps. 47% of them were always negative and therefore also not capable of distinguishing, but 43% of them had variable expression. So we focused on this 43% of variable markers to try and determine which could be indicators of cell health. And we did find variability in expression when we looked at all of the conditions, that is the different donors and the different cell culture conditions. We did see variabilities in expression of the markers, and this heat map indicates that. And when we looked at variability between the donors, if there was variability in expression of these markers between the donors, we found less evidence of donor variability, but we did see significant variability depending on how the cells were expanded in culture. And so what you see here is conditions A and B are on the left side of this heat map, and conditions A and B are on the right side, and condition C is on the left side. So we do see markers which distinguish the cells which had good quality from the cells which had poor quality. And so now we have a panel of markers which we're trying to validate in terms of their utility in predicting the quality of the cell preps and ultimately their utility as GMP markers. But what's important to note was that when we looked at those markers which are always positive and which don't distinguish between cells which are healthy and cells which are not healthy, we found that 4 markers which are very commonly used as quality release markers for MSC, that is CD73, CD90, CD105 and CD166, are always positive irrespective of the quality of the cell prep. And that's very important because these are the widely used markers in terms of quality control testing. And I wanted to focus for a minute on two of these, CD73 and CD105, because my lab identified these markers many years ago as being expressed on the surface of MSCs. So CD73 and CD105 are the antigens which are recognized by a panel of antibodies called SH2, SH3, and SH4, which were raised by Arnold Kaplan and Steve Hainsworth at Case Western Reserve University. My lab identified the antigens recognized by those antibodies as being CD73 and CD105. So we thought at that time that these could potentially be useful markers for testing MSCs. A number of years later, the International Society for Cell Therapy proposed a minimal set of criteria for defining mesenchymal stromal cells, and amongst those criteria, they suggested that MSCs should be, first of all, plastic adherent, and then they should express CD105, CD73, and CD90. I think now we have to question that these standards very, very strongly, and I think it's time to maybe change that, because the observations that we've made is that CD73 and CD105 specifically are not markers that give any indication about the quality of the cell preps. In fact, they do the opposite. They hide serious issues. And so in terms of marker selection for MSCs, and the important question that we need to address as we try to translate this field is that at least for CD73 and CD105, these are not effective markers of MSC potency for any target disease indication. They don't provide information on cell health, viability, or growth kinetics. In fact, they hide a lot of problems, and that they really should not be used as release tests, and specifically in GMP applications where the cells are being used for treating patients. CD73 and CD105 should really be stood down as MSC markers. One of the other aspects of determining the surface immunophenotype of these cells is that when you begin to understand the range of growth factor receptors, adhesion receptors, and so on that these cells express, you can begin to think about using this information for media development. So if you know the growth factor receptors, you can think about making cocktails of growth factors specifically to develop new defined media to remove the need for using serum. And so we've been doing that for several years now, specifically looking at ECM adhesion molecules, growth factor receptors, and molecules associated with proliferation of the cells, and then using this information as sort of a systems-based approach to develop new media. And we've had a great deal of success in this regard. So using essentially media consisting of recombinant growth factors only, we've been able to develop several. And here you see an example of an experiment where we looked at different combinations of growth factors. Several of these didn't work, but two of these, medium 7 and medium 8, performed better than serum-containing medium in this particular observation. And one of these media we've now taken forward to a GMP application as a fully xeno-free cell culture medium for MSC expansion. So we can address other aspects of the translational pathway that we need to look at. In the final moment, let me talk about some of the work we've been doing in trying to understand the mechanism of action of MSCs. What is the therapeutic, what are the pathways that are evoked when these cells are delivered to an injured tissue? We've done some experiments in the context of myocardial infarction, which are worth mentioning in this regard. And it's based on the idea that the phenotype of the cell delivered to the injured host is different to the phenotype of the cell that exists in a tissue culture flask. And we specifically need to understand that in vivo phenotype to understand the mechanism of action. The idea being that the cell, when it's delivered to the injured environment, is licensed, or its phenotype is changed in response to that environment. So the way to address this is to transplant the cells into the injured host and then retrieve the cells and try and understand how the phenotype of the cells is modulated by exposure to that injured environment. And we've done this in the context of myocardial infarction and also more recently in the context of the OA joint. So we put the cells in, they're labeled, and then at periods of time later, 3 days, 7 days, and 21 days, we retrieve the cells and try and probe how the phenotype has been modulated during the time the cells have been exposed to the injured environment. So this shows how we do that. This is an example of an injection tract of PKH26 labeled cells injected intramyocardially in the infarcted heart in rats. We can sort the cells by bulk sorting, and this is an example of the sort of separation we can get. So we can retrieve the hearts from these animals that have been treated with the cells. We can digest the hearts into a single cell suspension, and then we can do bulk sorting to retrieve those fluorescent cells. We can put the fluorescent cells back into culture, they still grow, and then we can probe their phenotype compared to controls, which were untransplanted cells from the same preparation, which were untransplanted. And to make a long story short, we've been doing this for some time now. We've done a series of gene expression comparisons between the retrieved cells and the untransplanted control cells. And we do see significant differences in terms of gene expression between those cells which were exposed to the injured environment and control cells. Some of those genes code for secreted protein, and this adds some support to the idea that paracrine mechanisms are important in this regard, that the MSCs, when they're transplanted locally, secrete factors which have an influence on the host. One of the particular molecules that we focused on in this regard was SPARKLE1, that's secreted cysteine-rich extracellular protein, which is secreted by the MSCs when they're exposed to the infarcted environment. And when we look at MSCs in culture, we find that they don't express this protein SPARKLE1. Cardiomyocytes also don't express this protein, but when the two cells are together in co-culture, then the presence of the cardiomyocytes causes the MSCs to upregulate expression of SPARKLE1. And then conditioned media from the cardiomyocytes also stimulates the MSCs to upregulate expression of this protein SPARKLE1. So SPARKLE1 is an example of how MSCs are licensed by exposure to the injured environment. And we've looked at what SPARKLE1 then does. In fact, it's anti-apoptotic to the cardiomyocytes. So when the cardiomyocytes are exposed to hypoxia, they undergo apoptosis, but this protein SPARKLE1 protects them against this. So SPARKLE1 is a cytoprotective molecule for the cardiomyocyte. So the cardiomyocyte stimulates the MSC to upregulate expression of this protein, and then this protein in turn protects the cardiomyocyte from death. And so this is the sort of cycle that I think we need to understand more of in terms of mechanism of action. And there's another part of all of this which is interesting, which is that we can then think about taking the protein itself and forgetting about the cells, the idea of a cell-inspired therapy. So we've done that. We've generated a SPARKLE1 patch, which we've sutured onto the surface of the infarct in these rats. And we find that this itself, the protein itself, without the cells, has an interesting therapeutic effect. And that's what you can see here. So here's the infarct size in untreated rats. Here's the effect of the empty scaffold when it's sutured to the surface of the infarct. And here's the effect of the SPARKLE1, the protein-impregnated scaffold. So we refer to this as cell-inspired therapy, and it may be, at the end of the day, that this kind of strategy will be more profitable than the complex business of preparing and delivering cells, living cells, in these models. So we've tried to address, number one, determining the clinical utility of these cells in properly controlled clinical studies. And we've also tried, number two, to address some of the translational obstacles which still need to be understood in terms of proper markers, more effective and useful markers, and then understanding the mechanism of action of the cells. And so let me finish there, and thank you again for giving me the opportunity to talk, and thank you for your attention.
Thank you very much. We have time for a few questions.
Hi, very nice talk. I was wondering, in the MSC field, there's been a lot of talk about what is the best cell, preparation to get in, and you touched on that, and you were talking about growth of the cells and maximizing the growth. when in fact, a lot of the literature talks about the more you expand the cells, the less they do. Could there be a way to maximize the function of the cell without maximizing the growth of the cell? Therefore,
So-
like doing some kind of selection in vivo? So you would treat the cells and then put them in a model of osteoarthritis, like one of the rapid models, and then find out what conditions you treated them in culture actually led to a better result in vivo.
So the point you make, I think, is absolutely correct. The idea of maximizing expansion of the cells is probably a mistake because it's just not going to work. You can't expand these cells at a very high level because they lose their phenotypic characteristics, they become senescence, they undergo karyotypic changes, and so I don't think it's possible to think about, very, very large scale expansion of the cells. This particular trial that I've been talking about is autologous, so the cells are expanded simply to passage 2. And we also find that the dose that we require is actually quite small, so the concept of sort of industrial scale expansion of the cells, at least in the context of what we're doing, doesn't seem to be necessary. We can think about rolling this out effectively on a small scale. Having said that, I do think you're right. We do need to understand much more about subpopulations, specific subpopulations of the cells, because what we're doing is very crude. We're still relying on plastic adherence as a major sort of biological characteristic of the cells that we use, that we take advantage of, and that's simply not good enough. So I think you're absolutely right. We need more sophisticated ways of understanding specific subpopulations of cells that could be used.
Right.
Hi, nice talk. 2 questions. One, in that trial, are you doing pre and post-op or pre and post-injection MRIs?
There is, yes, yeah. So there's 4 MRIs, pre-injection, and then up to 2 years.
Is that one of your outcome? What are you looking for in that?
So it's the protocol that's being used for MRI assessment is MOCART, and so the intention is to look at changes in the cartilage.
And then, there's strategies out there where folks don't expand the cells in tissue culture. They emulsify them and they do direct injection. Any thoughts about that?
I think it's not possible to say because there isn't the quality of follow-up that's sufficient. I mean, there's been a lot of talk about this, the idea of simply preparing a concentrate of bone marrow or fat and using that. It needs to be tested in a fully controlled clinical trial before we can say, and it just hasn't been.
Right. Thank you.
Could I ask you, you mentioned in that prior trial, you'd seen a very rapid decrease in pain in the patients. Could you comment about in your current trial whether that has been reproduced?
We can't say yet because the study hasn't been unblinded. But it's important to make a couple of points about that. So there has been a pain response which was really quite dramatic. However, the phase 1 study was open label and non-placebo control, and we do expect that there'll be a strong placebo effect in this. It's also worth pointing out that at least 2 other clinical trials which have been published, one in osteoarthritis and the other in disc degenerative disease, both also reported this interesting pain outcome.
Okay, thank you very much.